Good day to all. Good day to you, Professor Andrew. This is Shankar.
Good
Good day,
day.
sir. Sir, ma'am, I think we are good to go.
Yep, I'm happy to begin whenever you're ready.
Yeah, okay, sir. Ajil.
Hello.
Yeah,
Good
good
morning, everyone. Good morning, everyone. Warm welcome all of you to the second distinguished lecture program on behalf of Instrumentation and Measurement Society of IEEE SB NSSCE, which providing IMS chapter around the world with talks by expert and topic and interest and the importance of IMS community. The lectures are among the most qualified experts in their own field. We offer our members a plus a chance to interact with the lecturers. I hope you can enjoy the section. Now I welcome Ajil K. Babu, Chair of IEEE IMS SPC NSSCE for the welcome address.
Oh, am I audible?
Yes, you are.
Okay. Okay. Hi, friends. So, on behalf of IEEE IMS SBC NSSC, cordially welcome you to the second IEEE IMS distinguished lecture program in the section. We are grateful to you for taking out some time to join us at this great event. I'm extremely pleased to welcome those who have been serving our student branch for a long time and those as well who are here today. So, firstly, I would like to welcome the chief guest of for the event, Professor Andrew Taberner, physicist, bioengineer, and a professor at University of Auckland, New Zealand. From 2020 to 2002 to 2008, he was a postdoctoral associate, research scientist, and co-manager of the Bioinstrumentation laboratory at MIT. He has authored more than 140 referred scientific articles in journal and published conference proceedings, 117 conference abstracts and short papers, and published 28 issued US, European, and other patents. So he also received the 2014 New Zealand Innovation Excellence in Research Award. Welcome, sir.
Thank you.
Next, okay, thank you, sir. Next, I would like to welcome Professor Shankar Jayaraj, sir. He is an active IEEE volunteer serving as Student Activities Chair, IEEE Kerala Section, Academia Working Group Coordinator, IEEE Kerala Young Professionals. He is a recipient of IEEE Art and Young Professional Outstanding Volunteer in Academic Award 2021, IEEE India Council Outstanding Young Professional Volunteer Award 2020, and IEEE Kerala Section Outstanding Volunteer Award 2020. He is also volunteering as MOCNI, TIIO, Government of India, and as HIM operator, and also a trainer in following domains like 3D printing, robotics, process automation, amateur radio, and software-defined radio. Like so. Welcome, sir.
Thank you, Ajil. Thank you for the kind introduction.
Next, I welcome Dr. Vijita S. Ma'am, the student branch counselor of IEEE SB NSSCE, who is providing constant high energy to her ward till date. Welcome, ma'am. Next, I welcome Professor Nigel Binoisa, Head of Department Instrumentation and Control Engineering, NSS College of Engineering. He could not get here today for some important reasons. However, he has wished all the best for the event. I welcome him even in his absence. Welcome, sir. And of course, I welcome Professor Anjali S. Nayo, ma'am, who is the Student Branch Chapter Advisor of IMS NSS College of Engineering. Welcome, ma'am. And the whole participant panels who are eagerly watching the section live on YouTube. We hope you have a great time and keep enjoying the events as you always have. Thank you.
Thank you, Ajil, for your words. Now I welcome Glorious Vijida ma'am, Branch Counselor of IEEE SB NSSCE, to have some enlightening words about IEEE SB NSSCE.
Ma'am, your mic is muted.
Right. A very good morning. Swati, am I audible?
Yes, ma'am, you're audible.
Yeah, fine. Very good morning to one and all, and warm welcome to our next set of DLP programs. And welcome Andrew sir to this program also. And it is actually a pleasure for me to visit or sit and watch all the DLPs that has been conducted by my young blood at NSS College of Engineering. We are a sound branch that is there at NSS, and I think almost all my students are hardworking towards creating something that is very required for the students. It's completely a technical event that has been actually planned so far, and might be in a year when we sit out for a meeting and decide what is to be done for a year. Mostly the DLPs come into being because Corona has supported us in this at least. We are able to have all the DLPs online. So we are able to bring in faculty from the other part of the world also, so that the students can have a better interaction and know about the latest trends that is there in technology. So once again, I support and as well as congratulate the IMS team for their work. And I wish all the success to them. And I wish this is a very good event for us. Thank you.
Thank you so much for your valuable words, ma'am. Our beloved HOD Nigel Binoy sir of Instrumentation and Control Department couldn't reach here because of his personal affairs. Now, but he has sent us a video note. Now, listen to his supporting words.
Thank you. Very good morning to the respected chief guest, all the faculty, participants, and everyone present here. It gives me great pleasure to introduce the Professor Andrew Taberner, Associate Professor, University of Auckland, New Zealand, who is going to talk to us about optical sensing in bioinstrumentation. Bioinstrumentation is a subject in which we all are deeply interested due to its importance in medical area, especially due to this current pandemic situation. So Professor Andrew Taberner is a physicist, bioengineer, and professor at Auckland Bioengineering Institute, New Zealand. He was a research scientist and co-manager of the Bioinstrumentation Laboratory at MIT and teaches subjects like bioinstrumentation and measurement in biomedical engineering. His research interest includes the development of scientific and medical instruments for measuring tissue, structure, and function. So he is honored by the Best Teacher Award, the Sustained Excellence in Teaching Award, and the Innovative Excellence in Teaching Award. So, ladies and gentlemen, please join me in welcoming Professor Andrew Taberner. Thank you, Professor. So please take over this section. Thank you, sir.
Now I would like to welcome Professor Shankar sir, who is an active volunteer and serving as Student Activities Chair of IEEE Kerala section for addressing the stage.
Thank you, Abhita. I hope I'm audible.
Yes,
Okay,
sir.
so good day, everyone. So distinguished Professor Andrew Taberner, Dr. Vijita, Professor Nikhil Binoy, Akhil, Grishma, Swati, Badra, and all the vibrant student volunteers out there. So it's indeed a great pleasure, as well as I'm joining as participant also. Personally, I'm also working towards an oral cavity detector, especially non-invasive. So I'm looking forward to attend this session. So uh, talking about as a SAC of IEEE Kerala section, NSS Palakkad has been the most vibrant branch, one of the most vibrant branch in Kerala. And it includes a lot of section volunteer as well. So I'm really looking forward to attend this session, as well as the other student members of all Kerala who would be also fortunate to attend a quality program like this. And on this account, I wish all the very best for the entire team, as well as IMS student branch chapter of NSS Palakkad, to have a lot of discussion lecture programs to the entire student community of Kerala. Thank you, and over to you, Abhijit.
Thank you, sir, for your enlightening words. Now, let's have the privilege of listening to the distinguished lecture of Professor Andrew Taberner, the distinguished lecturer of IEEE IMS, who is a physics bioengineer and currently professor at University of Auckland, New Zealand. I invite Professor Andrew Taberner to take over the section.
Thank you very much for your warm invitation this morning. It is evening here in New Zealand on Saturday, same day as it is in India, of course. It's raining outside, but this is a nice way to spend the evening to meet some colleagues further around the world from us and to talk about the things that we care about. So I very much thank you for your invitation. I hope I can be heard, and I hope that you'll be able to see everything okay. Please, somebody let me know if there are any technical problems, and I'll try and do my best to fix them. So let me now just put my display up. Just give me one moment. So I hope now that you can see my screen. Can somebody please give me an indication that you can see the first slide of my presentation before I start talking?
Yes, sir, it's visible.
Okay, excellent. Thank you. So I won't be able to see the Google page while I'm talking, unfortunately. So please speak up if you need me to stop for some reason. So again, thank you for this invitation to speak. I'm coming from New Zealand, speaking from New Zealand, where I work at the University of Auckland. And in particular, I belong to a group that's called the Auckland Bioengineering Institute. I'm also the chair of the IEEE Instrumentation Measurement Society New Zealand chapter. So we in the Auckland Bioengineering Institute care about developing methods and techniques that help us to manage disease, understand our bodies, and generally make life better for all of us around the world. And we all know how important, don't we, measurement is in understanding the natural world around us and in building better devices, better treatments, and in this way, helping to improve people's lives. So my goal today is to talk about some of the methods that we have used and do use routinely in our laboratory to make optical measurements, so to make measurements using different methods of light. And it's those that I want to focus on today. And I'm just going to be covering 4 examples of different optical measurements in our lab. But first, as is always the case, I have to just very briefly do some advertising, and it's really probably not necessary given that many of you will be members of this society, but this talk is sponsored by the IEEE Instrumentation and Measurement Society. Again, we're a group of people all around the world who care about measurement, and one of the reasons for that is that by measuring things, we truly are able to say that we know something about them. And as Lord Kelvin, William Thomson, which was his real name, is often been quoted to say, to measure is to know. So we're interested in the science and technology of instrumentation, making measurements using now electronic devices. Of course, we have many different things, and one of them that you're taking advantage of today is one of our programs, which is the Distinguished Lecture Program. But you'll be familiar, I'm sure, with the development funding that is available and how chapters can obtain funding to support local activities. Our annual conference is the I2MTC conference. Now, of course, it's been very difficult for all of us over the last couple of years to participate in such conferences. They have to be done online rather than in person, but they're still worthy uses of our time and effort for interacting with colleagues. So you can see a list of the conferences sponsored by this society on our webpage. We publish IEEE Transactions on Instrumentation and Measurement, the magazine and the newsletter that is circulated to our members, the DL program that you're hearing today. But there's also many activities, of course, for students to network, to receive funding, best dissertation awards, student activity funding that can be used to support activities, and also for women in instrumentation and measurement throughout the world. So I encourage you to take advantage of all of these different things. I'm just going to skip through them. There are different awards and medals and activities that we have in our society that you can participate in. A society is only as good as its volunteers. So remember that the only way that these societies do us all any favors is by each of us contributing our time and effort to them. And so I thank very much the members of our society and of IEEE in general who are on this call today for the voluntary service that you give to helping us all to make life better for each of us. So just before I begin the technical part of the topic, I'll tell you a little bit about our country and where I am. I'm from New Zealand. I was born here. Of course, New Zealand is one of the last parts of the globe, really, to be inhabited by human beings. The indigenous population of New Zealand, the Maori people, came here somewhere between 800 and 1200 years ago. But many Europeans began to colonize the country about 200 years ago, and my ancestors came to this country about 150 years ago. Currently, I live in Auckland, which is New Zealand's largest city. I studied in New Zealand as a student of physics, and came to Auckland to do a postdoctoral fellowship at Auckland University, and then I was fortunate to win a fellowship to go to MIT in Boston, and I spent a few years there today. Some of the work that you'll see me present today was work that I began when I was at MIT. Auckland's a city of about 1.5 million people. It's this little, sorry, let me turn on my laser. It's this area here mostly that people live in. This is central Auckland. There are many people also in an area called South and North. And we're surrounded by two harbours on the north and south sides. We at University of Auckland, we're New Zealand's largest university with about 40,000 students. 2,100 of those are PhD research students, 2,600 are master's students. We're a comprehensive university, so we study all of the topics. But we also have two large-scale research institutes, and I am from this one here, the Auckland Bioengineering Institute. Again, bioengineering for us means using, well, our slogan is engineering better lives. So we think of ourselves as engineers who are applying engineering methods and insights and principles to studying life. And our particular focus, I suppose, is human life, although we do have some work with other animal species. But really our goal is to use the principles of engineering to better understand our own physiology in health and in disease, and use engineering to create better solutions for the problems that we face as human beings. So that's the overview of what bioengineering means to us. As I say, bioinstrumentation is the part of that engineering effort that is about measurement. So light's very useful in making measurements for a number of reasons. You can measure things about other objects, tissues, bodies, without having to touch them physically, except with photons, which of course don't make much impact on people unless they happen to be very high energy or in very high frequencies, like X-rays. So light's useful because it's non-contact. You can have single point measurements that can be conducted with single photosensors, or you can have area-based measurements, such as using cameras. And so light has had many different applications in all areas of instrumentation and measurement, but particularly in bioinstrumentation. So one example that I'm going to talk about today is stretchy length sensors, where we use light to measure the length of materials that can be stretched a long way. That's quite useful in instrumentation in the body when you want to measure the stretching of arms or the tension in muscles. Stretchy, compliant length sensors can help in that way. So I'll talk a bit about those. I'm going to talk about needle-free drug delivery and how we can use light in a very clever way to tell us about how well we are able to deliver drugs into the skin without using needles. And that's a very important tool for helping us to treat diseases that can be treated with liquid drugs. I'm going to talk about some other light-based techniques that we use for studying the contraction of muscles in the heart. And I'm going to be talking in particular about the mathematical method that we use that underpins that technique and allows us to use camera-based images to study heart muscles. But don't worry, it's a Saturday. We're not going to have any mathematics. It's going to be very simple. There's no maths in this talk. We don't need that on a Saturday afternoon or morning. And then finally, my final topic will be on measuring tissue polarization. So this is again another optical measurement where you can look into and through tissues in some cases. And the polarization that happens of that light, that occurs to the light as it passes through the tissue, can tell you something about the properties of the tissue. So I'm going to discuss that. There are many other examples that I could have chosen, I suppose, for this talk, but I'm focusing on these because we've done some unique work in this area, and I thought it will be of interest to an audience who may be bioengineers, but may also be from other areas of instrumentation. Okay, let's go. So this is a topic that has been 2 or 3 years, indeed, this ago. But we've got a couple of PhD students at the moment working in our lab, Alistair and Hayden, and Professor Paul Nielsen and I have worked together for many years. And so the work that we're showing you today is based largely on what we have been doing as a team. So this is a measurement technique that allows us to detect the length of a stretchable optical fiber. So what you're looking at here is a fiber that is made of an elastomer. It's a material that can be easily stretched. It doesn't take much force to stretch it. It's compliant, and it's also transparent. So it can be used as a light guide, like a normal plastic optical fiber, but a stretchable fiber. And by shining light into one end of that fiber and detecting that light at the other end, we can quite easily and quite well detect the length of the fiber. You can see here in this video recording, I hope, that white light is entering the fiber, but as it comes to the other end, the color of that light is further through the fiber. We can exploit that technique, and more generally, we can exploit the principle that light is attenuated in the medium with length in order to detect the length. And here's a visual representation of this happening. You're seeing here a video recording of one of the students pushing down on a piece of glass that is compressing a blue colored puck of an elastomer. And as it's squishing, it gets thinner, and so we can more easily see through it. When it is thickest, it's less transparent. So how can we make this into a length sensor, a stretchable length sensor? Well, one of the materials that we use for these light guides is polyurethane, and it has a refractive index of about 1.48, which is quite high. So if you surround the polyurethane with another medium that has a lower refractive index, for example, air, then the light that is traveling through that, in many cases, will be totally internally reflected. So you can now use the material as a light guide. And that's what we're doing in that example I just showed you. And so the intensity of any light that enters the fiber is attenuated by the fiber in accordance with the Beer-Lambert law. And so the intensity that makes its way to the other end of the fiber up here, where there is a photodetector, drops off with distance in accordance with this law. M is here the absorption coefficient for this particular material. I0 is the intensity of the light that is being shone in it. And so using this very simple equation, this is about as complex as the maths is going to get today, we can solve for x, the distance or the length over which the fiber has been stretched, by measuring the intensity that comes out of the far end of the fiber on the right-hand side. But it turns out, as you saw from the video of the stretched fiber, that attenuation is wavelength dependent. So blue light is attenuated far more strongly than red light in the fiber. And so that has the effect that the intensity of red light as you change the path length by stretching the fiber, the intensity of the red light doesn't drop off as quickly as other wavelengths. And so that's useful because now we can use either white light and shine white light into the device and look at the color change, or we can choose different wavelengths and perhaps use LEDs or lasers to shine light into the fiber and separate out the effects of the different wavelengths. So here you're seeing, this is in fact inverted. These two should be around the other way. The red light drops off at a different rate from the blue. And so we can now take the ratio of these two intensities by separating the colors from one another and use both of those pieces of information to determine X, the distance over which the fiber has been stretched. And you can do so by just simply taking the ratio of the intensities of those two measurements. Typically, the way we do this is that we use an LED as our light source, and we modulate that LED at a particular frequency. The red LED would be modulated at, let's say, 800 hertz. The blue might be modulated at 1100 hertz. And then at the photodetector, we can separate those two signals from each other, take the ratio, and that allows us to better detect the length of the elastomer. So here's looking at it in a slightly different way. Here's the same piece of material being stretched to different amounts. And that allows us to fit a combined Beer-Lambert law and detect the photodiode voltage, which here is red ratio to green, over a range of different lengths. So that's wonderful. We've now got a method for making a very soft and compliant material. We can stretch it over quite large distances. The strains that these materials can be exposed to are up to 500%. So you can stretch them 5 times their normal length and they don't break over that range of strain. That's quite remarkable. And we haven't yet built length sensors that work over that same range, but you can certainly stretch them by 100% and make a very useful strain sensor over a large length of displacement or strain. The other thing you can do, which I'm not going to show any results on this today, is but you can dye or color the elastomer by adding different colored dyes to the material. And those dyes then can also change the way that the light propagates through the material. So that can be very useful too, to specifically choose the colors of your dye that you add to the elastomer as you create it. And we make these elastomers ourselves using standard recipes. And you can then tailor the absorbing properties of the dye to interact appropriately with the color of the light that you are injecting into the fiber. So that allows us to then make an effective length sensor. This is just a really rough and quick measurement that we did in our lab of extending a fiber by a known amount and showing the predicted value. We haven't, this particular result hasn't been, we haven't measured the linearity, but you can see the sort of results that you can easily get with these fibers. What are we using them for? Well, we're using them in our soft robotics. So this is just an example of a robotic device that's been used in a exoskeleton for amputees to fit the prosthesis around a prosthetic stump, or the stump of an amputee, their lower leg, using actuated soft materials. So these happen to be pneumatic actuators, and into each of these actuators we are planning to insert these optical fibers, so they will be part of the structure, and that will enable us to measure the deformation of each of these actuators using a soft material with nothing hard in it. So we've got a particular device for doing that, but it's a form of our stretchable length sensor. Okay, so that's my first example of how we can use light in bioinstrumentation. The sensors and detectors that we use with those are just photodiodes and lasers or LEDs. They're relatively cheap, and you can make a length sensor that's relatively inexpensive but very effective. Okay, my next topic is quite different. So this is some work that I began, let's see, it's now 15 years ago at MIT in Boston. It's a needle-free drug delivery technique. And so, as the title says, needles are needless. You don't need to use a needle to deliver drugs into the skin. And so let me tell you a bit about how this works. This is a cross-section of a piece of skin. This could be animal skin, such as pig, or it could be human skin. A needle-free jet injection can be delivered or completed by taking a little, think of this like a sort of a syringe, but instead of having a needle on the end of it, it's got a very tiny hole. And so this is the plastic bit here, and there's a hole. The blue is some liquid, and the hole is only about 200 microns in diameter. So that's about the diameter of one human hair, roughly. It's a little bit more than for most of us, but it's about that sort of size. And if you pressurize the liquid to about 20 megapascals of pressure, a jet will come out of the hole at very high speed, 200 meters per second, and that jet will penetrate through the fat, through the skin, sorry, through any fat that underlies the skin and into your muscle, if you wish. And that can be as deep as 20 millimeters. And so the beauty here is that the fluid itself, the liquid drug, is a jet. And so it forms a very high speed penetrating jet that pierces into the skin. Now, how do we do this? We use electric motors. So this is an electric motor that I designed and built at MIT. We have smaller ones now that we're building in New Zealand. It's a linear voice coil motor. Very, very simple. It's a sort of motor that's in a loudspeaker, except loudspeaker motors don't move as far as this one does. So there are coils of wire that are in a magnetic field. You can see the magnetic field vectors radially protruding through the wire, through the coil. And by driving current through the coil, we can push a piston to squirt liquid out through the end of the injector and into the skin. Now, because this is an electronic device, we can control it. So we have position sensors on this to measure its position in real time. And we have a hardware control system that performs electronic feedback control on the speed of the drug. As you can see, it's bidirectional. We can squirt liquid out, but we can also draw back. And that allows us to suck blood out of the site where we have injected. So we've just recently published a paper where we have used this to perform a blood test. Now you'll notice also that the injector doesn't have to inject all of the quantity. It can do it in small steps. And that's very useful because it means what that reflects is that we've got very tight control over how much drug is injected. So one of the things about jet injection is that it is very fast. So you're looking here at a high-speed video recording of a jet injection into a transparent tissue. It's a transparent gel that we use as a model of tissue. So this video has been slowed down. This is all over in about 20 milliseconds. This video that you're watching, we're injecting black dye into transparent tissue. So here's where the jet comes in. And you can see the jet penetrates quickly and then fills in at that depth in the tissue. It fills the drug. It's pretty much centered on about this location here, but you can see the end of the fluid just penetrating slowly down into the gel. Now this also happens in in animal and human tissue. So this is in the animal. This is in pig skin, and you're looking here at an X-ray video recording. So over about, what is this one? This one's about 80 milliseconds. It's slightly more volume. My student James performed these measurements at MIT when he was visiting the MIT lab to conduct this experiment. So it's an X-ray video. You can see a rapid penetration and then filling up once we get to that depth. So this raised a question a few years ago in our lab. could the depth of this jet injection be tracked? Is there some way of measuring how far and how fast the actual drug itself penetrates into the tissue while we are delivering it? Can we measure that depth as we are delivering? Because if we could, then we could control perhaps the depth of the injection in real time. At the moment, we can control the depth of delivery in a jet injector by changing the speed of the jet. The depth that the jet penetrates into the tissue is strongly related to how fast the jet is. But if we knew exactly how deep the drug was as it was being penetrated into the skin, we could precisely, we think, control the actual depth of the drug during an injection. So that means, of course, that the method that we might use to do this has to be very fast. It's got to be able to give us feedback millisecond by millisecond, or even faster than that, preferably. So this was the challenge I posed to my student Karen, and this is how we did it. Well, we realized that the jet that comes from a jet injector looks a little bit like this. It's very thin. This is about 100 to 200 microns in diameter. This is a high-speed video of the jet in air. The jet's traveling from left to right. So you can see that as the jet emits from the jet injector, you get this very, very long, thin, slender, high-speed column of fluid. Now, in many cases, that fluid is transparent. It can be used then as a light guide. You can shine light down it. So once we realized that, we came up with this idea, well, perhaps we could, this is a cross-section of one of our jet injectors. Here's the fluid before it's injected, and here's the jet going down into the tissue. Could we put some light into it and just shine a beam of light down through the jet of liquid? It's very thin, of course, so we don't have much room to do this, but this is the idea. And of course, when the light travels down through this jet and it strikes the tissue, it's going to be scattered off that tissue and move and flash out through the media. So here's showing the pulses of light that we might squirt down through the jet, hitting the tissue and radiating back out to the surface of the skin. Perhaps we could detect that light on the surface of the skin. So that was the idea. So here's looking at it in a different way. Here is a block of tissue. You imagine this is some tissue on an animal or a human. Here's the injection site. And so the blue liquid is being squirted in, and the light that is being shone down with the liquid will strike the tissue, and we expect to see a pattern like this on the surface of the skin. If the light can propagate through the skin, through the muscle, through the fat. And so we built a device to test out this idea. So here's one of our jet injectors. This one's squirting, it's pressurizing the liquid across in this direction. And then the liquid turns 90 degrees and is squirted out through the hole, which is down here, and into a block of tissue. At the same time, we include a laser beam in here so that the laser is focused and aligned right down the instrument through the hole. Again, this is about 100, 200 micron hole, and into the tissue along with the jet. And then the idea was that we would use cameras, high-speed cameras, to see if we could visualize that light going into the tissue with the jet of liquid. So here's our laser, the motor is behind here, and here is a block of tissue with a couple of mirrors around it so that we can image the injection from a high-speed camera. Let's zoom in on this region. So here is a block of tissue from pig skin. That's the epidermis up here, the outer layer of skin. The dermis is just underneath that. That's a layer of fat. And then here is some muscle below here. So this is about 20 millimeters from here to here. So using our high-speed camera and this sort of setup, we were able to measure, to visualize the front of the tissue and the side of the tissue by looking at the side in this mirror. And we're able to visualize the top of the tissue to see how the injection, the light that is being injected into the tissue, makes its way to the top surface of the skin. And these are the video recordings that we took. So here we're looking at the front of the tissue, which is not really very interesting. This is the side of the tissue from the side mirror. And this is the top of the tissue, the surface intensity of the light during an injection. Now, the wavelength of light that we've used for this device is in the near infrared. So somewhere, I forget exactly the wavelength, but it was probably around 800 nanometers. Now, at that wavelength, light propagates fairly well in tissue. You can shine light through the tissue fairly easily at those wavelengths. So you can see here, this is the easiest way to visualize it. This is the injection point, and see when the injection starts, here, the light rapidly goes down into the tissue. So you're seeing that sort of shadow of the light as it propagates through, down with the jet and out through the tissue. At the same time, you can see what's happening with this on the surface of the skin. There is a change in the intensity pattern on the surface of the skin as we had hypothesized. So this was excellent. How can we use these two bits of information to determine the depth of the jet going into the tissue by measuring the intensity of this light? So Kieran did a whole bunch of analysis on these sorts of injections. Using the side distribution of the light, we have some information about how deep the jet is, because the center of this light pattern that we see will be aligned with the tip of the injection and where it strikes, where the light is then striking the surrounding gel or surrounding tissue. Sorry, this is not gel, this is pig skin. So the centroid gives us information about the depth of the injector. And at the same time, we can integrate the intensity, the radial change in intensity between the injection site, which is here, and we're looking at this top surface of the skin. We have information about how much the light is dropping off with the depth of delivery. So we integrate radially around that angle the intensity of light as a radial distance from the injection point. And so these are the sorts of results we got from a jet injection. Here is the distance of the center of the light as a function of time after we begin an injection at time 0. So very quickly, as you saw in the X-ray videos and in our video recordings of injections into gel, the jet very quickly penetrates to about 4.5 millimeters, and then it sort of slowly fills deeper and deeper into the tissue. That's exactly what we see in our transparent tissues and when we do our X-ray recordings. So that was excellent. That gave us a great deal of confidence that this measurement here was telling us about the depth of the jet in the tissue. Now, of course, you can't, if this was a human being, you can't see the side of the tissue during an injection, can you? All you can see is the top surface. And so what we want to be able to do is to know whether our image of the top surface can allow us to predict this, or more particularly, to predict the depth of the injection. Now, the surface profiles, this is the normalized intensity on the surface of the skin, was measured at every time point as you move along this curve. This is the depth of the center of the side. This is the intensity pattern on the surface of the skin. As we go deeper and deeper, the shape of this profile changes. So when the jet is first penetrating into the skin, most of the intensity is very near to the orifice. That's, I suppose, what you would expect. As the jet penetrates deeper into the skin, the light is going deeper into the skin and it scatters more. And so the shape of the light pattern becomes more spread out and more shallow. The intensity also drops off. This is normalized intensity, normalized to the intensity, I think, at must be at this depth here. So it's more noisy. So can we use this information to predict the depth of the injection? So I don't have time to talk about the details here, but we used a machine learning approach to try and train a system to correctly detect the depth of the injection using only the information from the surface of the skin. And so this is the result of the analysis we did. We used our knowledge of how deep the injection is from the side as our truth, and we trained a machine learning algorithm to predict the surface, to predict the depth from the surface image. And so this is a ratio of the two intensities that one sees from the side. And the solid dots mean that our prediction was correct. Circles mean our prediction was incorrect. And you'll see mostly these are solid dots, which means that this trained classifier was able most of the time to correctly predict whether the jet had entered the fat layer of the skin or had made its way down into the muscle. Now, the reason we were only trying to detect either of those two events and not actually measure the depth in millimeters is that usually in drug delivery, we don't actually need to know exactly how deep the drug is going, but we do want to know whether it's in the fat layer, where it will be slowly absorbed, or whether it's in the muscle, or whether it's in the dermis, just beneath the skin. So it's really, we only really need to know the layer of skin rather than the actual depth. So this method worked very well. And the way that we use this going forward in our future work is to use optical fibers to measure the intensity on the surface of the skin and very fast photodiodes to give us that information in real time. And then that will be fed to our classifier or to our machine learning algorithm, which will correctly, we hope, allow us to control the injection depth. Once you've got to the desired layer in the skin, you then slow down the jet so it goes no further. So that's an interesting application to jet injection that I wanted to touch on. Let me switch now to talking about camera-based measurements. And this work was largely conducted by these three people in our lab during their PhD projects. So we have developed an algorithm that we use in many of our measurements to determine very, very small movements of objects or very, very tiny differences between images. And it uses a method that is often referred to as digital image correlation, but the particular technique that we use is a Savitzi-Golay differentiator. I don't have time to talk about, and I promised I wouldn't talk about maths, so I won't talk about how this works, but I'll just show you what it can do. It's an extremely sensitive way of determining the distance that particular regions of an image move from frame to a tiny fraction of a pixel resolution. So we can resolve down to about 1,000th of a pixel. What we typically do is we take an image and we divide it up into sub-images. We use this technique to detect the displacement between the image frames, and then we use some phase information to resolve that displacement to a millipixel. And the benefits of using this particular approach, which you will find in this journal paper, are that it is incredibly robust, it's efficient, it doesn't take a lot of computational power, and it doesn't require any intrinsic markers. And I think it is the most accurate method that I have yet seen, or sorry, the highest resolution method that I've seen published. Let me show you how we've used it in a couple of applications. First of all, looking at heart tissue. Muscle has a visible structure. If you take a piece of muscle and put it under a microscope, this is what it will look like. The contraction of this particular muscle happens from left to right, and the muscle is activated by these stripy proteins called sarcomeres. So here is a video recording of a piece of heart muscle being twitched electrically over and over. This is what your heart is doing right now. Of course, it's doing it much faster than this. This has been slowed down. But you can see all the movement of the tissue in this sample. So we want to be able to, in our research, measure the strain. How much movement are we getting in this tissue, and how does that change over time? The way we do it is we take these small samples of tissue and we collect images like the one that you were seeing, and we stitch them together. So if you imagine this is the microscope looking at this, we can move our sample backwards and forwards using motors in the microscope, collect all of our images, stitch them together, and now we get a video recording of the overall motion of this slender piece of tissue. This little actuator, little motor, is the subject of another talk, distinguished lecture that I've given many times. But these are muscle fibers that are taken from the heart, and you have some in your heart of this type. They're again about 200 microns from here to here and only about 2 millimeters long. So we've got all this enormous amount of information in these images. Can we measure things from it? Well, yes, we can. So by splitting up this image into many sub-images, and each of these dots represents the center of a sub-image, we can now use our image registration technique to measure the displacement of each of these dots during the movie file or across frames. So here's an example of us doing that. We're looking at the X displacement computed from our algorithm and the Y displacement. The X is more interesting because that's the direction in which the muscle is shortening. So you can see that the red indicates that this end on the left is moving to the right, and blue indicates that it's moving to the left. These dots, of course, are not real. They're just the position of virtual markers that we have added in our software to visualize what's happening. This is very useful information. By looking at the distance between the dots, we can now compute the strain using this method. So here's the strain field. There's very high positive strain at this end of this fiber, and that's because this muscle is not very healthy at this end. This tissue has been damaged. There's high negative strain at the other end because this part of the muscle is actually very strong. As the muscle contracts, it's contracting and producing force on the right, and the force is pulling the tissue on the left. And that's why we get positive strain on the left and negative strain from the muscle itself on the right. And there's also information in the Y direction. This is a single cell taken from a heart. And here again, you can use this technique to measure cell shortening. This particular shell is stuck on the left-hand side to a cover slip. It can't move. But you can see the right-hand end of this little motor contracting to the left. And our method being able to track the motion in this cell, despite there really not being very much that we can see with the naked eye, this algorithm's able to detect that motion to a tiny fraction of a pixel. So that's really quite marvelous for us. We can look at how this motion changes along the length of the muscle fiber and how it changes over time, because these are video recordings. We can select a region in the sample and track its position over time. So this, as a twitch occurs, this particular sample of tissue moves in one direction and then back down again. It goes around this loop, sorry. And so it's moving mostly in the X direction because it's shortening, but there's also some Y direction movement. Of course, we can also compute the strain. The strain field in this myocyte, single muscle cell, is quite uniform. That's good, it's healthy. That means it's contracting almost the same amount all through the muscle cell. And we can quantify that during the twitch. So what other things can you use this method for? Well, it's not just, of course, muscle cells and microscope images. There are other applications in bioengineering, but there are also many other applications all across engineering of, using techniques like this. Here's an example where we have used it to look at the arteries and the veins on a human neck. Now, the reason for doing this is that as people age or develop diseases of the heart, that can cause quite large deformations on the surface of the skin. I remember seeing this on my own father. When he was in his 90s, he had a heart disease, and his neck, when he was lying back in his hospital bed, was moving quite profoundly as the blood pulses through it. So we took a video recording of a human neck, and we used our image registration technique to measure the deformation of the skin And this line here represents the line, I think in this case it was the carotid artery, or it may have been the jugular vein, I'm not sure which. But by looking at the deformation of all of these pixels or these areas of the image, we can measure now something about the deformation of the skin and something about the blood that is pulsing through it. So here's an example of just 3 points. These are the vectors that show, according to our image registration algorithm, how far the skin is moving over time during the heartbeat. So you can see that we're only measuring a maximum of 6 pixels of deformation, which is why it's so hard to see with the human eye. But our algorithm can measure to a millipixel, and so we can get these lovely waveforms of deformation from these video recordings. So these are 3 example images of using our method in blank and comparing our results to using a laser interferometer sensor, which was extremely expensive. And we're just using a relatively inexpensive webcam for this measurement. You can see the sort of quality of our method compared to using a very highly expensive and precise measurement. So that's incredibly useful because these waveforms, these pulse waveforms, tell us a lot of information about the heart. And it's this particular thing here. These are heartbeats. You can see here the ECG signal, time synchronized. This pulse here is referred to as a dichrotic notch. It appears in the pressure waveform, and it's because of the valves in the heart. And when these valves become diseased, there is quite a profound change in this timing of this pulse with respect to the ECG and the main pressure pulse. So we are now using this information in a human study to see if we can use this optical technique to detect disease in diseased humans. Here's another example of using image registration. This time it's for doing 3D reconstruction. So now on this device, we have 4 cameras looking at the surface of, this is actually not skin, it's a piece of silicone. And we can reconstruct the shape of that silicone using this image reconstruction technique or image registration technique. That's very useful, but it's useful particularly when you want to do things like measure the deformation of skin, perhaps during surgery. So this is actually a very exaggerated measurement of cutting a piece of rubber dam and tracking the motion of that rubber dam and the shape of the dam using a 4-camera stereoscopy system and using image registration. We've done it on rubber, we've done it on pig skin. This is a piece of pig skin with some spots on it, but usually we don't need to add any spots on the skin to track it because the features that we all have on the surface of our skin are sufficient. And another application of this technique has been on the lung. The image on the left there, which I'll just run again, is an X-ray, CT X-ray of a rat lung, showing all of the air structures within that lung. Well, we can measure the external surface of one of these lungs as it is being inflated. So this is a rat lung. This is just one image, but we have a 12-camera system examining 12 different views of the same lung as it is being inflated. So here's the 12 cameras positioned around the lung as the lung is inflated. Because we are measuring the deformation on the surface of the lung tissue, as we inflate it, we can measure the deformation from the camera images and the shape of the lung. And that gives us an enormous amount of information about how this sort of little balloon is being compressed by the incoming air and distending. And no one's ever really been able to make these measurements in this way before. At the same time, we use another optical technique, which I don't have time to talk about, optical coherence tomography, to look inside the lung at the alveoli. The alveoli are the very small sacs that we have in our lung in which the gas exchange between the lung and the blood, the air and the blood is being implemented. So that's a wonderful technique. Okay, my last topic, thank you for your patience. I hope I'm not going too much over time. I don't think so yet. This is a final topic that we have only just published in the last 2 or 3 weeks. And this is about using an optical technique to look through biological membranes. So we've built this really quite interesting instrument that allows us to take a piece of tissue and put it in this device and stretch the tissue using a bunch of motors at the same time as examining the tissue with a polarization analyzer that we have built. So here's the piece of tissue. This is actually pericardium. That's the sac, the balloon that's around the heart. That tissue is very valuable. It's used to create artificial heart valves for humans, but it's usually gleaned from young calves, so bovine, bobby calves, are often used as the source for this material. Other species of animals can be used for these types of measurements and for these types of heart valves. So this is what a pericardium tissue looks like. It's transparent to different wavelengths of light. Again, mostly near the infrared. The redder end of the spectrum it's more transparent, but this is just white light shining through a pericardium. Now, using our image registration technique, we can measure how much the tissue deforms as we stretch it. So you're looking here at a scalloped piece of tissue that's being pulled by these fibers here, which are connected to linear motors with force sensors on them. So we're stretching the tissue and we can measure its deformation using image registration. The reason we're doing this is to help people who need transcatheter heart valves and that surgeries that are made of these biological materials, and sometimes more frequently now being made of artificially generated biologically mimicking materials, so materials that are grown in the lab and perhaps seeded with cells. We want to be able to make these measurements on them. The reason that we use an optical measurement is that the materials that biological tissues are made of are very often polarizing, and that's predominantly because of the collagen within them. So if you can measure the polarization properties of tissues, you know something about the structure of the collagen within that tissue. And that's very, very interesting because you don't have to cut the tissue up in order to know where the collagen is and in what direction. So I won't go into the physics of it, but there are many optical properties that can be measured using this approach. We've built a thing called a Mueller matrix imager. Now, for those who are interested, these are called Stokes vectors. And so they tell you completely about the polarization state of light going into an object. And the Mueller matrix is the transformation matrix that can tell you everything about how the polarization state of the light that goes in, which is this Stokes vector, is transformed into the light that comes out. So without going into any detail about what this Mueller matrix actually is, we measure the Mueller matrix directly using our polarimeter, and from that we can fully characterize the optical properties, polarization properties of the tissue. And that's very valuable, as I'll try and explain now. So basically, our device has a light source. It's an LED. At the moment, just one LED. We have a rotating polarizer filter, a quarter wave plate that shines light into the sample. So this is called a polarization state generator, PSG. Here's our biological sample. And then on the other side, we have a polarization state analyzer. And by rotating these objects, we can shine light of different polarization states into the sample, and by using this, analyze them by collecting a bunch of images. So the sorts of images that we collect, I think 16 images in order to measure one piece of tissue or one field of view. But we can also measure maps of the polarization state of this tissue over large areas. This is a 4x4 array of one piece of tissue. We've overlaid it with a grid. And this is the polarization, one of the polarization properties of that tissue measured by our polarimeter. It's the linear retardance. There are many other different properties we are measuring, but linear retardance is the one that's of most interest because the linear retardance of the tissue, again, I don't have really time to explain what that is related to the collagen structure and the collagen direction. So in this way, we can create maps of that tissue. These vectors are showing the direction of the collagen fibers, and the color here shows the sort of density of those fibers by the amount of retardance that they are causing. That then allows us to predict the directions of the fibers or measure them, and potentially the stiffness of those fibers without cutting up the tissue. And that's very useful for engineers who want to make heart valves out of this tissue. So that's how we do it. Here's just finally some examples of measurements we've done while stretching pericardium. These are the 16 different images that we create. And from that, these are once we've measured the different elements of the Mueller matrix, we can now watch these as this fiber is stretched over time. So this is a video of that fiber being stretched. Some of these Mueller matrix images relate more closely to the properties of interest, linear retardance, but we have to decompose the matrix. We do all of this mathematics, which I promised I wouldn't talk about, so I'll skip. And that then allows us to predict linear retardance in these tissues as they are being stretched. So here's an example of a fiber, a piece of tissue being stretched only in one axis. And you can see that the retardance is changing in that tissue as it is stretched. And that is because the collagen fibers are being realigned predominantly along the length of the stretch. The fast axis of the realignment is an optical property, and the circular retardance is also rotating as the material is stretched. We're hoping to use this next to, again, be able to relate the optical properties we measure to the natural collagen state within the tissue and use it as a predictive tool. But as you can see here, as we stretch this piece of tissue by 15%, you can see how these vectors, which represent the linear retardance, are being stretched and aligned with the direction of the tissue. Okay, thanks everyone for your attention. I think I've talked for long enough. I'd like to acknowledge the wonderful students in our lab who come from all around the world. Many come also from New Zealand, were born in our country, but I would say probably, I think at the University of Auckland, 60% of our students are international. And so I think that's probably reflected in this this photograph. Shubham was Indian and he was visiting us from your country. Many others come to us from Iran. There's quite a few Iranians in this photograph, and they're all wonderful students to work with. So thank you very much for your attention. I shall stop talking now and pass back over to the people who are running this session. Thank you so much.
So, there are some questions from YouTube live stream.
Okay.
So I will post it in the chat box.
Okay, if you can put them in the chat box, I'll try and go to them now. I might just have to end my show. Okay, so I hope you can all still hear me. There's a question here, even though it's needleless, won't it be painful with the high pressure injection? Actually, well, there's certainly a sensation with needle-free injection, but the pressure that's used in a needle-free injector is about the same as the pressure of the needle tip on the skin, of course, and the pain sensation is different. It's not as painful. There's many studies published about needle-free injection. Ours is not the only needle-free injector, by the way. It's not as painful, judged by most people, as having a needle. Pain is one aspect, though. The other aspect is just people are afraid of needles, and so needle-free injectors, of course, avoid the problem of people being afraid of the needle. The fact that a needle injection takes a lot longer is another issue, because once the needle's inserted, of course, the nurse or the doctor or whoever is delivering the injection can sometimes turn the needle. I know that's some of my friends have been complaining about that when they've been getting their COVID injections here in New Zealand. Sorry, their COVID vaccinations, not injections. So no, the answer is that there is a sensation from a needle-free injection, but most of the studies show that it's not as painful as receiving the equivalent needle injection. Part of the reason for that is that the jet of fluid is actually thinner than the smallest needle that is used to deliver drugs into the skin. The jet is only between 150 and 200 microns in diameter. It's very difficult to get an injection needle that small. That's smaller than about 33 gauge. So I hope that answers your question. What's the difference between prosthetics and prostheses? Well, nothing really. I mean, they use the term is used in different ways. The prosthesis I was referring to is the prosthesis that is put on the stump of a human when they are having a prosthetic limb. If they have lost a limb, perhaps due to whatever, people routinely in the Western world lose limbs due to diabetes, but of course other people in other countries might lose limbs due to landmines and things like that. The prosthetic limb is really what I was referring to. We're using our soft actuators, our soft actuators to conform to the shape of the limb, and because they're actuatable, we can move the air in and out of them. We can relieve the pressure that is placed on the leg. So prosthetics often also refers to things like artificial noses and ears. So no, there's nothing really different between a prosthetic and a prosthesis. The impact on the optical coefficients for the needle-free injector. I suppose that question is for necrosis and edema on optical coefficients. Yeah, that's a really good question. Don't know the answer to that one. Necrosis on this, I haven't seen any work that I can think of on how those vary in tissue death. I mean, the tissue that we study is, yeah, no, it's a good question for diagnosing tumors. Yeah, I don't know. That's a really good question. Nothing that we have done in our lab in that area, and I'm not really familiar with the literature. Thanks for a great question. Sorry, I can't really answer it. Wow, that's a hard question. The question is, could I suggest the cutting edge research of optical biosensors, MEMS, AFM microscopy, and photonic biosensors? I don't think that I can, because I'm not, I mean, I review papers in this area. There's certainly a lot of work being conducted around the world. The proceedings of SPIE are a good place to look in biomedical optics, which is where we tend to publish. You'll see in there, of course, the most recent work that's being done. All I can really comment on is the sort of areas of focus in our lab, and we're not really developing biosensors that take advantage of AFM and MEMS. We are using optical techniques for detecting blood dilution, which is not a topic that I've talked about here. And that's very important for us. But of course, AFM uses optical techniques, of course, to measure the deformation of the AFM cantilever. So I'm sorry, I'm probably not giving you very useful information here. All I really know about is the work that we do and the papers that I review in this area. but there's obviously a lot of work going into photoacoustics is an area that's taken a lot of recent development for measurements in tissue, but those are more structural than sort of biosensing. So yeah, I'm sorry, that's the best I can do. Well, for us, the question here is, what are some of the most challenging topics of research that include membrane curvature and imaging optics? There's some very interesting work going on in our institute to examine, to use time-resolved techniques, super resolution microscopy, to look at membrane curvature and see well beyond the diffraction limit of the optics. So that's some really interesting work that I have seen. The author or the researcher in our institute who's doing a lot of work in this area has named David Baddeley. And so super resolution microscopy is a very powerful technique for looking down to sort of the nanometer level using traditional optical wavelengths. And I know that he's been looking at membrane curvature using that technique. So that's the thing that pops into my head when I see that question. Can I talk about the relevance of imaging techniques like photoacoustic imaging and diagnosis of tumor? No one in my group is using photoacoustics yet. I think there is some other work being done at the University of Auckland. Certainly seen some really neat stuff at meetings of the IEEE Engineering and Medicine and Biology Society in photoacoustic imaging. I'm not sure that they were specifically using it for tumor recognition, but I remember seeing a great keynote. I can't remember now the author of that keynote at the IEEE EMBS meeting. I think the last one that was probably held in person, or certainly the last one I went to, and I can't remember where that was now. But yeah, that's certainly a hot area. We have not done photoacoustics. This is where you combine, of course, optics with ultrasound or tissue or methods like that. But that's not an area that we have done work in directly. But I think it is very relevant. Some of the imaging that is enabled by that technique is quite fantastic. I don't know how much clinical acceptance those methods are getting yet and how far the commercialization has gone. But certainly the most recent stuff that's come out over the last 10 years has been quite astounding at the sort of resolution and information that can be gained with those techniques.
So I think that's all from the.
Sir, I think Shankar sir is having a query. Sir.
Okay, what do I need to do to answer? You can go ahead. Yeah, of course.
Okay, so thank you for the talk. So my question is mainly into the photosensory part. So let's take a spectral sensor chipset. So in that we will have pre-different light sources. But for a particular set of application, we really need to look into a particular set of wavelength. It will be like the manufacturer doesn't have a particular capability in the particular range. So we need to adopt filter to do that, optical filters to achieve that. So what is your suggestion in developing a product that is in line with what we need to do, especially in the range of wavelength? I hope.
Yeah, I think I understand your question. So you have a problem where you need a particular wavelength, but there aren't matching manufacturing technologies that allow that wavelength to be produced. Is that really what you're saying?
Yes, exactly. So we need to rely on this to do that.
Yeah.
That's
Well, of
actually
course, yeah, you've of course got to have a broadband light source that you can extract the wavelength from in order for that to work. I mean, in all of our applications, we usually use LED and laser light sources for the work that we do. in portable devices because they, we can generally find the close enough wavelength to what we want. Is this a problem particularly related to a portable device? Because of course you can use a halogen or xenon or whatever light source to have a broadband and filter it. So yeah, I guess it depends on how precisely you need the wavelength that you want, but filtering and filtering a broadband source, or sometimes you can move the move the central wavelength of a photo emitter by modulating the current. That can be sometimes done, and certainly you can do that on photodetectors as well. By for example, on a photodetector, you can move the sensitivity, the wavelength of peak sensitivity by modulating the reverse bias that you put across the phototransistor or photodiode. So yeah, it's an interesting question. How do you? Yeah, I suppose it depends on the context. In the lab, usually we can find a filter that works for the particular application that we have. It just depends, I suppose, on the bandwidth of the light that you need and how tightly you need to control that.
Yes, we need to have like a response of 20 nanometer.
No, yeah,
So that is actually quite challenging.
yeah, it is.
And by using filters, it's actually very difficult that we won't be able to get a particular response, especially since we are detecting the light source and we are doing a ratiometric analysis. But
What
I'm not
about
sure how well
what
we
about
will
your
be able.
fluorescence technique? Is there some way that you can excite the wavelength that you want in a narrow band?
Okay.
I don't know. I mean, that might be a silly suggestion, but
Okay. So we have to work on that. So anyway, definitely I will get back to you.
yeah, I
Because
mean,
that is
it could be possible. I don't know which wavelengths you're talking about, but if you can find a fluorophore that has a very narrow emission spectrum and is centered on the wavelength that you want, potentially that could be an approach to generate the wavelength that you need. But of course, anyone who is a manufacturer of light sources will also have access to those same fluorophores. And if the manufacturers can't do it, I'm not quite sure whether, I mean, that probably suggests that those fluorophores are not available, or if they are, they might be very difficult to use.
As we are connected with the manufacturers, what they seems that they won't be able to produce that particular component because it's highly costly to develop
Right.
that component. So that's actually quite challenging. So we are trying to have 6 wavelength identified, and we will work towards narrowing it down to 2, and then we will see. So hopefully, when this process all in place, definitely I will reach out to you for suggestions.
Sure. Yeah. I mean, the other approach, rather than using filters, is to use diffraction.
Yes. Okay.
To split the spectrum up and be a lot like a monochromator.
Okay.
But that's probably not very efficient either.
Okay. Anyway, this we are just opening for all. That we need to make the, as you said earlier, better day for tomorrow for humanity. So anyway, definitely it's wonderful to have you here, and thank you for the team for inviting me. Also, it's actually fruitful for me as well as for my product development. Thank you, and thank you, Professor Andrew.
Yeah, thank you. Thank you for your attention.
Sir, we have one more question in the chat box.
Yeah, so the question is, can multispectral imaging discern the spectral reflectance from surface and layers beneath the highly scattering tissues? Yes, the answer is yes. We tend to, in most of the work that we've done on skin, we have been deliberately trying to look beneath the skin to subsurface features. But yeah, absolutely, multispectral imaging can give you information about the layers in highly scattering tissues. So multispectral imaging, of course, for those who I'm not quite sure, is when you're using different wavelengths in the light source and looking at the reflectance and absorption and scattering of those wavelengths. in information that conveys about the layers of the skin that are being illuminated.
Well, I think that's a conclusion for the Q&A session. Now we can go on to the vote of thanks. Dishma or Abita, yeah.
Thank you, sir, for the section. It was really a great and informative section. I hope everyone enjoyed the section. Now I welcome Grishma J, Vice Chair of IEEE IMS SB NSSCE, to deliver vote of thanks.
Thank you, Abida. Good afternoon to one and all. It's such an honor for me to get the opportunity to thank you all dignitaries on the behalf of IMS SPC and SSCE. First of all, I extend my gratitude to Professor Andrew Taberner to take out his time and efforts towards this distinguished lecture program on optical sensing in bioinstrumentation. I'm sure all of us had a great time and got an excellent coverage on this topic. So once again, thank you so much, sir, for sharing this much of knowledge with us.
You're very welcome.
Thank you. Now, I would like to acknowledge our gratefulness to our Honorable Principal, Dr. Suresh Sir, for his support towards conducting this program. I mention our deep sense of appreciation to our very own Dr. Vijida Miss, Counselor IEEE SP and SSCE, for her guidance and motivation. Thank you, ma'am. I also wish to express my gratitude to Professor Shankar J, Chair of SAC IEEE Kerala section, for his presence and volunteering us today. Now, I may like to express our sincere thanks to Professor Nigel Binoisi, Head of the Department, Instrumentation and Control Engineering, for his supports and contribution towards us. Further, we are grateful to Professor Anjali S. Nair, Staff Advisor, IMS SPC NSSCE, for her presence and support. I also thank Dr. Sudha ma'am, former principal of NSS College of Engineering, Palakkad, for her presence today and support shown towards us. Also, I thank all the staff members of Instrumentation Control Department and all chapter advisors of IEEE SBC and SSCE for their enormous cooperation towards the event. Also, I am immensely thankful to Ms. Abida for her efforts towards anchoring this event today. As we all know, an event like this cannot happen overnight. It requires a lot of planning and coordination, which start rolling many days ago. We have been fortunate for having such a dedicated crew who works hard towards the successful culmination of this event, and I'm thanking all of them. Also, those who were a part of this event directly or indirectly. Thank you all once again. Thank you all for the great success of this event. Thank you.
Thank you, Grishma, for that words. That's end of our section. I hope everyone enjoys the section. Once again, thank you all for your active participation. Special thanks to Andrew sir. And thanks for the viewers in YouTube. Once again, thank you all. Wishing you all a good day. Thank you. Thanks for the viewers in YouTube. Once again.
Thank you all. Stay safe.
Thank you.